Welcome all the Duke alumni and friends to the Forever Learning Institute seminar series. It is my distinct pleasure to welcome you to what I hope will be a very fun session. And along this theme of immunity and regulation of the body and protecting the body, we thought that one of the key things that we want to address is aspects where the immune system can go awry. Either when it's trying to protect us from pathogens or when it's just trying to do its own set of normal activities within the body. And so we have three speakers from both the Department of Immunology and also from Biomedical Engineering. Dr. Saskia Hammers is an assistant professor and a Whitehead scholar. Dr. Ed Miao is a professor of immunology, also a Duke Science and Technology Fellow. And Dr. Joel Collier, who is a Theodore Kennedy Professor of Biomedical Engineering. And so the themes that we're going to hear about today have to do with topics related to the basic functioning and regulation of the immune system. You're going to hear about a subset of cells that are the most important function is to regulate the immune system. And you're going to hear about how they're controlled in terms of numbers and activities. Then you're going to hear from Dr. Miao, who's going to tell you about cells that commit suicide as a mechanism to defend themselves against pathogens, and that this has to be done in a correct manner, otherwise things go awry. And we'll be closing with Dr. Collier, who's going to tell us about how maybe we can leverage the immune system and coax it into making therapeutics that will help us control immune responses. So with that, we're just going to go ahead and just jump right in with Dr. Hammers.
Yes, thank you so much for this opportunity to introduce myself to you and some of the research that is going on in my lab. So today I will actually tell you two short vignettes that are related to research in my lab that center around regulatory T cells. So what are regulatory T cells? They are very potent suppressors of immune responses. And a dysfunction of regulatory T cells actually has been associated with many disease states, and that's due to their really powerful and potent immune regulatory role. So most famously, if there are defects in regulatory T cells, that can lead to autoimmune manifestation, but regulatory T cells are also involved in promoting transplantation biology, pregnancy outcomes. They can interfere with allergies, chronic infections, and acute infections, and also interact with our host microbiota in our gastrointestinal tract. So, but what are regulatory T cells and where are they placed within our immune system? So regulatory T cells are part of the adaptive immune system, which is this really small part of our overall immune system that's comprised of B and T cells. And unlike the vast innate immune system, which is sort of pre-programmed to react to common broad categories of pathogens, the adaptive immune system is highly specific to each particular pathogen that the body can encounter. To illustrate that, during the course of an infection, here illustrated by a virus, it's first detected by cells of the innate immune system, just as broadly as a virus entering our tissues. Those innate immune cells then get activated, and then they can migrate to the body to specialized lymphoid structures that are also known as lymph nodes that you might have heard of. And in those lymph nodes, those dendritic cells or innate immune cells can then interact with B cells and T cells, the cells of the adaptive immune system. And only T cells that are very specific to the infecting pathogen, in this case, the specific virus, will get activated. They will mobilize and multiply and then help with the clearance of the virus. So one of the features of the adaptive immune system that I've highlighted is how specific for a pathogen they are. But they also have another feature that is that they can form long-term memory. So after the virus is cleared, some of those activated T cells will actually become memory cells that then, upon secondary encounter with the same pathogen, can react more quickly and help clear the infection sooner. And that's actually the basis of vaccination. So how do T cells achieve this high pathogen specificity? The answer lies into lies in the nature of its T cell receptor, creative name choices, I know, that actually detects pathogen-derived material. So the T cell receptor doesn't recognize the whole virus or the whole bacteria, but it actually recognizes small bits and pieces, which we refer to as peptides. And they can, and the T cell receptor, as it recognizes those small peptides, one of the interesting things about the T cell receptor is that the specificity of the receptor is actually not encoded in our genes, but the receptor is generated through this very fascinating mechanism called VDJ recombination, which you can just imagine as a combinatorial building block assembly, which leads to a theoretical diversity of unique T cell receptors that each of us could have of more than 10 to the 15, an unimaginably large number. None of us have actually 10 to the 15 unique T cell receptors circulating through our body, where the estimated diversity of T cells within our body in mice and human is somewhere in the range of 2 million to 20 million. But thinking of those numbers, that's still between 2 and 20 million different specificities or different pathogens our T cells can recognize, which is quite remarkable. As I mentioned, though, the specificity is not actually encoded. So that means that the T cell receptor itself is agnostic to whether the peptide that it recognizes is derived from a pathogen or whether the peptide it derives is actually derived from ourselves. So there's a high risk of T cells getting activated by our own proteins, right? And that is something that we want to avoid, because if our own proteins would activate our T cells, that would lead to autoimmunity and could be very, very damaging or deathly to our bodies. So there are layers of protection in place. And the first layer of protection is actually happening when T cells are developing in a specialized organ, the thymus. So you can view this process really as like a quality control checkpoint where the T cell receptor is tested for its functionality. First, it's assured that it actually works. And then it's assured that all the T cell receptors that bind too strongly to peptides that are derived from ourselves and therefore could precipitate autoimmunity are removed. And this quality control process is really efficient and also quite stringent, because only 5% of the cells that enter the quality control process actually make it out as functional T cells in the end. However, even though this process is already quite stringent, it's not sufficient. And there's a second layer of protection in the form of one of my favorite cell types, the regulatory T cell. And regulatory T cells are T cells themselves, but they're a specialized subpopulation that can dominantly suppress autoreactive T cells in various ways. And that's in a process that we refer to as peripheral tolerance. And those regulatory T cells really sort of were rediscovered in the late 90s and early 2000s. And in the past 20 years, we've learned a lot about them. And one important thing that we've learned is that, for example, in humans that have a genetic deficiency, which leads to them having no regulatory T cells, that is actually leads to a fatal autoimmune disease that already manifests really, really early in life, known as IPEX syndrome. So those regulatory T cells are potent suppressors of immune responses that employ many different types of mechanisms of how they interact with other immune cells, but also with broader tissue physiology and organismal physiology. And so I would like to highlight two short vignettes of work that my lab has been doing in the context of regulatory T cells. And the first story that I would like to briefly share with you is centered around the question of how the size of the regulatory T cell pool is actually determined. So I've told you that regulatory T cells are very, very powerful in modulating immune responses. I think it's somewhat intuitive that the number of regulatory T cells must be set to an optimum number, because if we have too many of those powerful suppressors, if we might not be able to mount a sufficiently strong immune response to clear a virus or help eradicate a tumor. On the other hand, if we have too few or none, we are at heightened risk for developing autoimmune diseases. So, I was very interested in understanding when the size of and how the size of the regulatory T cell department might get set. And I reasoned it might happen during their development in the thymus. So like other T cells, regulatory T cells develop in the thymus, but one unique requirement that's specific to regulatory T cells is that they need to, that they're dependent on this soluble mediator called interleukin-2, and to fully become a regulatory T cell. But interleukin-2 is also a limiting resource for this developmental process. And so we were really interested in understanding what cells within the thymus are actually providing this limiting resource, and therefore would determine the size of the regulatory T cell population. And so my lab really likes to use mouse genetics because they offer us tools to study the immune system that would be really hard to do just in the human system. But we've used a mouse that basically marks all the IL-2 producing cells permanently with a fluorescent reporter that allows us to identify those cells within the thymus. And we used parallel approaches, including newly arising technologies like single-cell RNA sequencing. And with all the data that we generated, we could identify the IL-2 producing cells as actually being self-reactive CD4 T cells themselves. So that led us to propose a model where it's actually the self-reactive T cell pool that scales the size of the regulatory T cell compartment through the provision of IL-2. So if we have very few self-reactive T cells that make it through this quality control process in the thymus, there would be low amounts of IL-2 and a small population of regulatory T cells. If there are many self-reactive T cells that make it through this selection process, there would be a lot of IL-2 and a lot of regulatory T cells. Therefore, it's always ensured that the size of the regulatory T cell population actually matches the population of autoreactive cells that they're supposed to suppress. And in a second short vignette, I would highlight a completely different aspect of regulatory T cell biology, and that is their role in promoting tissue repair after tissue damage. So my lab is really interested in understanding how regulatory T cells that reside in tissues actually detect damage to the tissue. And we reason that it might be through receptors that can detect damage-associated factors. And I just want to highlight one brief story from my lab. So we like to use a mouse model of multiple sclerosis in my lab. And one of the things that we noticed was that the damage-associated protein IL-33 is elevated highly in mice that showed signs of multiple sclerosis, so active disease. And this was curious to us because regulatory T cells express high levels of the receptor to detect IL-33. So we wanted to test whether this detection of IL-33 or sensing of damage by regulatory T cells was actually relevant in this disease setting. And so again, we used a genetic mouse model that allows us to specifically remove the receptor for IL-33 from regulatory T cells. And then when we compared the disease severity in those mice, it became clear that the regulatory T cells that couldn't sense the damage in the tissue anymore were not as capable as keeping the disease in check. So the mice had worse disease outcomes in this multiple sclerosis model. So with that, I'd like to wrap up my part of the session. I hope I've given you a glimpse into the wonderful world of regulatory T cells and how they are important for human health and interact with many different body systems in our body. And I'm happy to take questions in the end. But before that, I'd love to pass on the baton to my colleague in the department at Miao, who will tell you about research that his lab does. Thank you.
Thank you, Saskia. So my lab is, I'm going to tell you about new concepts in the cell death field that my lab is developing. We're proposing that caspase, I'm going to tell you about, allows cells to complete their bucket list before they die. So we're interested in studying pathogens, bacterial pathogens that invade our cells and try to replicate inside of them. So for example, we're going to focus on Salmonella today, that uses a molecular syringe to reprogram our cells to allow it to invade and then replicate inside the cell. This is obviously disastrous, but we have sensors in the immune system that can detect the activity of this syringe and enact a solution, which is to get rid of the infected cell. Once it's been reprogrammed, that cell is an agent of the enemy, and we need to just kill it through regulated cell death, such as apoptosis and pyrotosis, which I'm going to talk about today. So what is pyrotosis? Pyrotosis is the name for the inflammatory or feverish nature of the falling, pyrotosis. This occurs when sensors that detect this syringe activity activate a protease called caspase-1. That then cuts a host protein, one of our own proteins called Gasdermin D, activating its ability to form a pore. Then that pore opens up and the cell swells, and then the membrane eventually ruptures and the cell dies. This is a video here of macrophages with a red dye in the media, and they're about to undergo pyrotosis. You can see that dye can't get through, but when the membrane ruptures, it's explosive cell and rapid cell death. This video occurs over the course of 45 minutes or so. So very dramatic and efficient cell death. We had studied this previously in macrophages, but recently the field has been interested in other cell types that may activate this caspase-1 pathway, including in the intestine. So if our intestine is a hollow tube, intestinal epithelial cells line the top of the villi like this. And when the caspase-1 activates in these cells, what they do is first they extrude, spit themselves out of the monolayer, and then they die after they've done this. And this is a challenging process because, and I'm going to call these IECs, intestinal epithelial cells, are essentially sewn together to their neighbors to make a really strong barrier. So they have to unsew themselves and squeeze themselves out. This process takes energy. The cell that's going to extrude has to disassemble its microvilli. It has to then reassemble its cytoskeleton and squeeze itself out of the monolayer. And then finally, at the last stage, it has to undo those zippers that attach it to the neighboring cells. So we can study this in mice. We'll be showing pictures of histology in the intestine with extruding cells. And we can also study it in tissue culture in organoids, which are essentially hollow spheres of intestinal epithelial cells, where the inside is like the inside of the tube of the intestinal tract. This is a video from one of our colleagues' labs, Russell Vance's lab, where they're showing this extrusion process. So here's one of these spherical organoids, and each little cube in here is a cell. Watch over here, where they've stimulated the caspase-1 pathway in all these cells, but the very first cell to get going is this one, and you can see it, bloop, this one, bloop. They extrude themselves into the lumen. Give this another minute or so, and the entire thing will go red, as every single one of these cells has been triggered. But it's a nice video to show that when you activate the caspase-1 pathway, extrusion occurs. But if you think back 3 minutes ago, I mentioned that caspase-1 also opens this Gasdermin pore, which is going to cause the cell to swell and pop. So these two events, the cell is on its way to death, but it needs to complete this extrusion process before it gets there, and it needs to be alive to finish the extrusion process. This only takes 10 minutes. You got to be alive for 10 minutes, but pyrotosis can pop a cell within 1 minute. So we are adopting a phrase from the Bucket List movie, where Morgan Freeman and Jack Nicholson's characters unfortunately have terminal cancer diagnoses. They decide before they kick the bucket and die, they want to, they've got a list of things they really want to do in life, and one of them is see the pyramids. We think that our own cells, they know that they're going to die once that caspase is activated, but before they die, they have some things that they need to finish. We're calling this the cellular bucket list. In the case of these intestinal epithelial cells, they have to finish extrusion. So we're developing this concept from our studies of this protein caspase 7. I mentioned pyroptosis and apoptotic cell death. Caspase 7 is one that seems to be involved in both arms and might be useful in both of these death pathways, but it was long, for 27 years, no one knew what caspase 7 did. So Kengo Nozaki, a brilliant postdoc in our lab, was studying this extrusion model in mice. These are mice infected with salmonella, and these are sections of their intestine. And you can see that the infection triggers individual IECs to extrude, to pop out of the monolayer and jump into the lumen. When Kengo looked at caspase 7 in these cells with an antibody that only binds the active form, you could see that they're brightly staining for this green caspase 7 signal. So they're activating the caspase, and Kengo found they also need this caspase to finish this extrusion process. In a mouse that doesn't have the caspase 7 protein, now instead of the cells extruding as an individual, they seem to be dragging all of their neighbors with them in these very abnormal extrusion clusters. So something is going wrong in this process. You can painstakingly count those. So going back to the organoid model, when we trigger extrusion in all of these cells, when we trigger the caspase 1 pathway in all these cells, you can see that the red dye will rush into all of these cells as they simultaneously all extrude and fall into the middle of the sphere. And you can look at this over time. In a caspase 7 deficient organoid, this happens faster. They turn red faster. They're dying too fast. And then we think dying too fast and failing to extrude. And so the mechanism that we've discovered is that caspase 7 is tying into a basic long-known membrane repair pathway, which is to use this protein called acid sphingomyelinase to repair a membrane. If this is a cell, when a pore opens up, ASM is triggered to be exocytosed and spit out onto the surface of the cell. And there it generates a copious amount of this lipid ceramide, which is a classical membrane repair pathway. When it cleaves the head group off of this lipid sphingomyelin to make ceramide, because it's of its shape, it causes the membrane to invaginate and spontaneously endocytose. And when you put that pore inside the cell, it no longer allows the water to rush from outside to make the cell swell and pop. So this is going to buy cells the critical 10 minutes that they need to finish that extrusion process. Putting this all together, what we've shown is that caspase 1, when it's activated, detecting salmonella, activates both the pore and caspase 7. The pore triggers this exocytic event, and it's also the conduit for caspase 7 to leave the cell to meet ASM outside the cell, to cleave it and make it ridiculously effective at making massive amounts of ceramide, much more than it would normally make to repair pores. And this ridiculous amount of ceramide that is generated can repair the ridiculous number of gas German pores that caspase 1 is creating. So you can see evidence for the generation of this ceramide lipid in this process. Here's one of our, actually 2 extruding cells in a wild type animal. And you can see here we have stained both for ceramide, beautiful red stain, right in the same cell where what is in white this time, the active caspase 7 is staining. And you can see the neighboring cells are not activating caspase 7 and they're not generating ceramide. But if you look in our caspase 7 deficient mice, here's one of these abnormal clusters. They don't make any ceramide to repair those gas German pores. And when we mutate ASM in a mouse so that it's no longer cleavable by caspase-7. These also do not make that ceramide. So they have their normal ASM, can make a little bit, not even detectable by this antibody. And caspase-7 in white here is being activated, but failing to get to this individual extrusion and failing to make the copious ceramide that is being generated here. So in conclusion, we think that cells have a bucket list of things that they need to do before they die, before they lose all metabolic activity. And in intestinal epithelial cells, the cell is opening gastrointestinal pores, which are going to de-energize it and make it pop. But before it does, it needs to let go of its neighbors. Otherwise, it's going to be a corpse stuck in the monolayer next to all these other living cells, and that's going to trigger them to probably die and fall out with it, but still tied to the cell that was supposed to be gotten rid of. So caspase-7 simultaneously activates a repair program to buy about 10 more minutes of life. Eventually, those gastrointestinal pores are going to overwhelm this repair pathway, but only after the bucket list is completed and the cell has extruded. So this was all done by Kengo Nozaki and Vivian Maltez in the lab, did an earlier part of this project. With that, I will turn it over to Joel Collier.
Thanks so much. It's so fun to be here and to talk to you all today. We're going to switch gears a little bit and talk about antibodies specifically. Just a few disclosures. So I want to focus on a very specific part of the immune system that I think we all have a reasonably good feel for what they do, and that's antibodies. I think we all appreciate that when we vaccinate, antibody production are one of the main ways that your body is fighting off pathogens. But also in recent years, antibodies have become one of the main forms that we produce pharmaceuticals, right? So if you watching TV, you see many ads these days for drugs that are actually antibodies. So anytime you see MAB on the end of the name of a drug like Humira or Taltz or Xolair over here, these are all antibodies that have been produced in a factory in bulk, and they're delivered to a person to have a pharmaceutical effect. So Humira is for knocking down a particularly inflammatory molecule that leads to diseases like arthritis or Crohn's disease or inflammatory bowel disease. Taltz is an antibody that interferes with another protein that's involved in the pathology of psoriasis. Xolair, you can see this man is ecstatic to be dancing through the garden, buoyed by these antibodies that are flowing behind him. So Xolair is a antibody actually against another antibody that is central to allergic diseases like allergy. And it's been an absolute revolution in the way we're able to treat disease. Our ability to produce over and over again specific antibodies that have these therapeutic effect have absolutely revolutionized pharmaceuticals. The biggest money-making drugs right now, the most high-volume drugs, are all antibodies engineered in this way. So they've been an incredible boon. However, they do have some shortcomings. To give you a few specific examples, so one of the most highly utilized antibodies out there are known as blockers of a protein known as tumor necrosis factor, TNF. So that's AbbVie's drug Humira. Remicade is another version. There are several of them that fall into this category. And for patients that for which it works, it's been life-changing. However, to start with, about 10 to 30% of patients don't respond at all to these drugs. And then over time, they may sort of have an initial response, but then over the course of weeks, months, years, the drug stops working. And that happens to another 23 to 46%. If you dial down to a specific example in Crohn's disease, by the time you add in folks that never responded in the first place, those are known as primary non-responders, and folks who stop responding eventually, those are known as secondary non-responders or loss of response. You have less than half of folks who are even able to keep using the drug in a useful way. Even though they're incredibly lucrative for the pharmaceutical industry, they're really quite expensive for the patient or the patient's insurance. $3,000 per syringe, around $77,000 annually. And they require to be almost always delivered by a needle. So either you go into a care facility and receive an infusion, or you're injecting them yourself. And so that has led us and others to think, are there ways that we can achieve these therapeutic molecules, antibodies, but then do it in a way that is more cost effective, that is less prone to side effects, less prone to loss of response, more easily delivered, easily delivered equitably across the world. You could imagine it's quite challenging to ship and to store these very expensive syringe formulated pharmaceuticals to low resourced parts of the world. And so, as an alternative to what is known as passive immunization, where the antibodies are made in a factory, produced, and then delivered over and over again to the patient, what if we were able to produce those antibodies in the patient themselves, right? So we deliver a much smaller, more easily transportable, more equitably distributable material to the patient, and allow the patient to then go on and make those antibodies. So the antibodies are going to go off and do the therapeutic work. If we can do this, there are fewer injections needed, less risk of losing response because the patient becomes sensitized, and it's a more long-lived response. And so this kind of work falls into the category of what we are calling as a subfield that's really popped up just in the past few years, a subfield of immune engineering. And this is a interdisciplinary research area. So I work in the biomedical engineering department. My background is in material science, and can you? Wait, hang on one sec. Sorry, can you still hear me? I had a quick glitch on my.
We can hear you fine.
Okay, just wanted to check. I had a funny.
But go back to full screen.
What's that?
Your slides are not full screen.
There. Are we back?
Perfect.
Okay, thanks. So I work in immune engineering, right? My background's in material science and protein science, and it's combining those types of engineering approaches with immunology to develop new therapies to engage the immune system, both for protecting against pathogens. There's a huge amount of immune engineering done in that context, as well as treating other diseases for which we don't have good treatments yet. The problem as an engineer, you'd want to understand the system that you're engineering with, try to understand the rules. This is a map of a very, very small slice of the immune system. This is just one signaling pathway of what are known as toll-like receptors that sense damage in the immune system. And so the actual full immune system to map out is even quite a number of orders of magnitude more complicated than this. And so it becomes really quite challenging as an engineer to think about how we might put many different signals together into a technology to elicit a specific response that we might be interested in, say, production of a therapeutic antibody. And so to simplify things, I like to analogize and think about, well, why don't we think about the immune system with this circuit diagram? This is a circuit diagram of a sound mixing board. So if you'll sort of allow me to keep going on this analogy, I like to think of the immune system as a combination of many different components, right? And to achieve the right therapeutic efficacy for whether you're doing a COVID vaccine, an anti-cytokine vaccine like what I'm talking about here, anti-cancer immunotherapies, one has to dial the settings to achieve the proper mix, right? And some of the things that go into the settings include what kind of T cells are involved. They can be different phenotypes, Th1, Th2, Th17. I'm not going to go into too much depth with all the different immunological terminology. It can be quite a morass. There can be different kinds of antibodies, IgG1, 2, IgE, that's the allergic one, IgA, that's ones that happen in your mucosal tissues like your lungs and your GI tract. How strong are the antibodies? How tightly do they bind to their target? Are they monoclonal, like the ones I talked about, like you'd produce in a factory? Are they polyclonal, a bunch of different antibodies that can bind the same target? And it kind of goes on and on. There are all these dimensions of how you would want to set those sliders. And so in our group, what we try to do is boil those down into individual components and then assemble them together into materials, into nanomaterials. And so we work with what are known as supramolecular materials. These are molecules that we can build in a modular fashion and then assemble together to bring together some of these signals that I'm talking about. What you're seeing in the middle of the slide is an electron micrograph where we have nanofibers that are about 20 nanometers in diameter and a few hundred nanometers long. We are talking 1,000-fold smaller in width than a human hair, even a little bit smaller than that. So they're really quite small, but they're studded with some of the molecules. This is an HIV vaccine that I'm showing here that we want to raise responses against. We synthesize all these in our lab. Shown here is a automatic peptide synthesizer. And so we'll have a domain that allows the fragments of the proteins to come together into a fiber. And then we'll decorate it with different epitopes, different fragments from either a pathogen or from a target that we'd like to raise an antibody against. And we might formulate them with other molecules that increase the immune response, adjuvants. And we can do this in a modular fashion so that we can tune some of those sliders. For an active immunotherapy, remember the goal is to produce antibodies that can go off and mitigate a protein that may be having a bad effect in the body, say a cytokine. But when it comes to prompting the body to produce those, we don't really want much else to happen immunologically. We don't want, say, T cells to go off and kill the cells that are producing those antibodies. We want a very focused, sort of thread the needle kind of immune response that is only producing those antibodies, right? So antibodies, no T cells, and we want to do it in a way that is non-inflammatory. So what a graduate student has done recently in my group is to take fragments of a protein known as tissue necrosis factor, TNF, and attach part of that to the self-assembling component. These, when you mix them in buffer, will arrange themselves into this long molecule that has the epitope, the target. When we raise an antibody against this, it will react against the cytokine of interest. By putting it in a long fiber, you have many copies of that epitope on the surface. It makes it into a particle. Collectively, these things heighten the response to the immune system. And then we can also add what's known as a T cell epitope. In 10 minutes, I have to gloss over a lot of the immunology, but this allows T cells to help produce those antibodies. We are in the process of testing these in preclinical models, mostly mice, but also rabbits and at times non-human primates. And we are finding that we're having good therapeutic efficacy. For example, when a mouse has a rheumatoid arthritis-like disease, its paws will swell and become red. And this is how severe the disease is after the induction of the disease. If you treat it with a monoclonal antibody, like against TNF, you will delay the onset of the disease and reduce its severity. Our immunization, in which the mouse is producing its own antibodies against TNF, will have also likewise delayed onset of disease and reduced severity. We're also doing this in a whole range of situations. Another one is inflammatory bowel disease, where if a mouse is given inflammatory bowel disease, this is done with, they have a chemical in the drinking water that irritates their colon. And so they have weight loss and they have disease severity. So plotted here, and you give it to them in a cyclic kind of way that replicates the disease. When we immunize them with our immunotherapies that will raise protective antibodies, their weight loss is much less, and the disease severity is less as well. And another way that you can measure the severity of disease is the length of the colon. When someone has inflammatory bowel disease, the colon actually shortens, and we can protect against that with the immunotherapies. So to sum up, I hope I've given you a taste for the kinds of things we can do with the immune engineering. We can raise antibodies that can go off and do immunological work in a way that hopefully is a more distributable, equitable, cost-effective, safe, broadly efficacious way. It's an alternative to passive immunotherapies that have brought us incredible distance, but not all the way in treating diseases like rheumatoid arthritis, inflammatory bowel disease, psoriasis, and others. This is some acknowledgements. Many of my students have been contributed to this, and we're mostly funded by NIH. So thank you. Looking forward to discussing and having some questions.
Excellent. Thank you, Saskia, Ed, and Joel, for wonderful presentations. A lot of thought-provoking ideas there. And I'm going to ask people to put the questions together in the chat, and I'll try to collate them and find patterns that I can go ahead and ask from the speakers. So maybe while that is going on, I can start asking a few questions, and maybe I can start with you, Saskia. You described very nicely how Treg cells put a brake on the immune response. Are there instances where pathogens can take advantage of that and perhaps use Tregs to control how the immune system reacts against a pathogen?
I mean, I'm sure they are, because the pathogens are always one step ahead, right? So I think thinking about the immune system in general is I think useful to frame it in an arms race, right? So whatever we come up with in terms of defending ourselves, pathogens are faster in mutating and adapting, as I think all of us have really learned during the COVID pandemic over the past couple of years. And they will come up with strategies that will outsmart our immune system, and then it might take a little bit longer to adapt. I think the good news is the way our immune system is designed is it's designed in multiple layers, right? We have adaptive immunity and innate immunity, and they're sort of layered on top of each other. And then there are cell types that can have overlapping functions too. And I think that is the reason why, we can persist in a world surrounded by viruses and bacteria that, are trying that could potentially do us harm. So I think the layering and sort of the network aspect of the immune system is what can protect us. But yeah, there are always chinks in the armor of our immune system, and that's a constant battle. But yeah.
Okay, as questions come in, some things will begin to emerge. But in the meantime, maybe I'll ask something from Ed. This bucket list, is it something that is specific to epithelial cells of the intestine? Or do you think this is a theme that happens everywhere, like your airway epithelia or your endothelium, as pathogens try to cross through blood vessels?
Yeah, I think we think it's something that happens in every cell of the body that is on its way to die, and that every different cell type has its own bucket list. So obviously macrophages don't need to extrude, but they may need to have accomplished some other task on the way to cell death. We have data in other models where we have pathogens infecting the liver, where hepatocytes we think are undergoing apoptosis. And we think before they lose all energetics, they have to finish the processes of apoptosis, which is essentially ways to take the cell apart. And we're going to classify that as also another bucket list. Finishing apoptosis is a bucket list that you must complete before the cell loses metabolism and energetics.
And Joel, there's some questions coming from you related to the monoclonal antibodies and the nanoimmunogens, how long they last, whether they've been in therapeutic use at the time, at this moment.
Yeah, best we can tell, they last on the order of days to weeks, but are cleared within a few weeks. We certainly can't detect them anymore. With that said, your immune system does have a way of hanging on to very, 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 very tiny quantities of antigen for longer than that. there are very, very few molecules left when that process is underway. So as best we can tell with the detection limits that we have of looking, we'll label our materials and then watch them clear over time, and we can sort of extrapolate down. And it looks like it's about a 2-3 week lifetime within the body. I saw some other questions on the are there any monoclonal antibodies in clinical settings? Now, many of them, right? There are many monoclonal antibodies that are in use for treating inflammatory disease, for infectious disease, right? Monoclonal antibodies against COVID. I'm not sure what the approval status of those are right now, but a large fraction of the pharmaceutical industry right now is based on monoclonal antibodies. Estimated financial advantage between current large molecule therapeutics. So there are a lot of factors that go into what the price of a pharmaceutical is, including the development cost and patent positions and a lot of things that don't specifically relate to how much it costs to build it. However, with that caveat, I would say that you need to generate far less material for an immunization than you do with monoclonal antibodies, which are going to be needing to be provided again and again and again and again throughout the lifetime of the patient.
I think probably what ends up happening, a theme that I'm seeing in a lot of the questions is aspects of autoimmunity, aspects of degeneration that may be happening as a result of a hyperactivated immune system. So maybe this touches both on kind of aspects of our use, Saskia and Joel, is when one starts tinkering with the immune system, what are maybe some of the unintended consequences that may be happening from long-term stimulation of certain pathways or the dysregulation of regulatory systems like Tregs? Maybe that you guys can comment on that.
Yeah, I think it sort of ties into my earlier answer that, the immune system is incredibly interwoven, and it's also incredibly well balanced, right? And sort of if you push too much for too long on one end, this balance will be disturbed, right? And like the consequences might not show up where you think they might, right? Because I think that's what we're learning in recent years too, sort of how layered also and how integrated the immune system is in so many responses, including like neuronal function, right? This is something that we didn't really anticipate, but the communication networks are much broader and much more intricately linked. And yeah, I think trying to focus on one cell type and then just looking for effects on that cell type, even from a therapeutic standpoint, will not be sufficient, just because it will affect many more parameters that can be incredibly hard to detect, and I think is a challenge when we think about therapeutic interventions.
Joel, do you have any comments about
Sure, yeah,
of immunity and how that may be perturbed?
Yeah, to add to that, I think what we are trying to do right now is, at least speaking for myself as an academician, is to try to build the toolbox for both turning up and turning down immunity, for shaping immunity, right? And we're not, tomorrow away from being able to do that clinically, right? We're talking about sort of a new way of thinking about immunological disease. And so we're adding, tools to the toolbox to do that. Right now, we've done well at the augmenting part, right? So we've gotten pretty good at being able to raise the antibodies that will go off and do the immunological work that'll knock down some of the pathogenic cytokines. We've done this I only showed you sort of the tip of the iceberg that we've been doing in our group. The tool that I'd like to spend some more time thinking about and some more time working is the brakes, or are the brakes. How do we tune down a very targeted immune response, right? Is it another antibody against a specific target? Is it engaging B cells directly? Pruning away specific B cell subsets. I think you have to have a full toolbox to really do this, in clinical practice. So we're not one step away. We're a few steps away from that. But I think if we can do that rather than producing, the molecules externally, and coming in with a very sort of limited kind of there's only so much you can do with that sort of approach. If we develop this way of thinking about managing immunological disease, then sort of all of them are sort of within our grasp.
So one question that came up, actually a direct message, and something that's becoming pertinent, certainly to me, that as we age, our immune system changes. And maybe you guys can comment briefly about how much work is being done on the aging immune system, and how some of the therapies and how the regulation of the immune system may be different in an aging population. Because a lot of these therapies are directed to things that manifest themselves sometimes later in age.
Yeah, that's something that we've actually been starting to bring into our thinking recently with inflammaging, right? So especially, so to give one example, older individuals don't respond to vaccination nearly as well as younger ones do, right? So for example, COVID vaccines don't generate quite as robust a response as they do in younger individuals. And so in some cases, that's because of the sort of more inflammatory character of their immune system. Some of the cytokines that we're actively trying to knock down, like TNF, actually work against good vaccine function. So we are actually trying to put together some experiments where we see if our pre-vaccination with anti-TNF, anti-IL-17, anti-IL-1 beta lower those cytokines and then allow an aged individual to respond better to a vaccine.
Anything to add, Saskia, Ed, about aging inflammasomes, aging Tregs?
I mean, I would also highlight that it's true both for the aged population, but it's often also true for sort of like the early life population, thinking about like babies and young children, right? Where immune responses are also quite different from what we usually sort of assume a normal adult population looks like. I think so at both ends of the spectrum, the immune system functions quite differently, and there's still a lot of space to explore. I think there are nice studies coming out, even with mouse models. I think one big caveat that we as a field need to address, I think, is that we often study aged mice, but don't take history of infections into account, right? And that's probably one of the major drivers that shapes an aged immune system is repeated exposures to different pathogens over lifetime that all of us have experienced. But the mice in our colonies hopefully haven't because they're kept in a clean environment. But so accounting for that in our animal studies, when we're using those as preclinical models, I think is also something that we as scientific community need to pay more attention to.
Okay, so we're running against the hour. There was a lot of questions related to COVID, neuroinflammation, vaccinations, and that could be a whole bunch of sessions onto themselves. So I wanted to finish by asking you very quickly, maybe you can tell us what do you think is the big challenges out there in your own fields, the big questions that need to be answered in the next 5 to 10 years?
I think in our field, one of the questions and challenges is understanding how when different cell types in the body are undergoing regulated cell death, how those morphologies may be different, how different pathways, I've talked about pyrotosis and mentioned apoptosis as kind of parallel pathways. There's interconnections between them. How do cells know which way they're going to go, what morphology they're going to end up in the end, and how does that the destination affect the overall immune response? One of the more recent discoveries is that cells can be programmed to switch from apoptosis to pyrotosis by expressing different gas German proteins, and that may be applicable in areas like cancer immunotherapy by switching a tumor from being more silent and apoptotic to being more inflammatory and pyrototic, and that might drive the immune system to attack it rather than ignoring it.
Saskia?
And that's just one cell type.
Yeah, I mean, I think in the field of regulatory T cells, it's sort of like we know that they exist, we know that they're powerful. We still honestly don't really understand how they actually suppress the immune system. And that's been, we learn more and more modes, but whether there is sort of a unifying principle behind that is really unclear. I think that's what people had initially assumed. It might or might not be the case. I mean, it has created a whole field of research that, a lot of people are working on, and it's clearly important and interesting, but I think seeing whether the field unites or sort of branches out into sub areas will be very interesting to see in the future.
Great. And Joel?
In less than 1 minute,
Yeah.
to me, the main challenge is that everyone's immune system is different. And clinically managing one person's disease versus another, I think, requires development of a whole new range of tools that we have, plus the wisdom and clinical experience to guide someone through use of those tools at an individualized basis. I think until we have the human and technical tools to do that together, we won't be able to treat each individual's immunological issues.
Wonderful. Thank you. I want to thank the three of you for wonderful talks, thought-provoking, and I hope everybody in the audience enjoy the conversation.